Hello, and welcome to the third in our 4-series webcast on advances in statistical genetics and the importance they may play in the insurance industry. In this third webcast, we're joined by Professor Cathryn Lewis, Professor of Statistical Genetics at King's College London, to talk about the results of the RGA-funded research that has been carried out at King's College London. Over to you, Cathryn.
Thank you, Peter. It's my pleasure to talk through the results of our research study with RGA that we've been carrying out over the last year. This is a project that was carried out with our research team at King's College London, Paul O'Reilly, Jessie Maxwell and Beatrice Wu. The aim of this study was to use the data from UK Biobank to estimate risks of mortality and morbidity. And the access to UK Biobank data is very protected by restrictions that they've put into place for the participants in this excellent study. And this is why it's a collaboration with academia, because only those of us at King's College London had access to the data. So the aims of this study were twofold. Firstly, they were to look at how we could predict risk of morbidity and mortality across different disorders using data collected in UK Biobank. And then, primarily, was to see how much genetic information increases the predictive ability of those models. So is our current knowledge of genetics informative enough that it gives us useful risk information? So the UK Biobank is a massive UK study of 500,000 people aged between 40 and 69 when they were recruited into the study, which happened from 10 years ago. And they've been followed up after that time, looking at different disorders that they developed. And of course, we can then use the baseline information that we have to predict risk of disease and model that. So there are 3 different strengths of UK Biobank that we're going to use in this study. So first of all, it has wide and deep data, a lot of information on 500,000 different people. It has follow-up across the years following their recruitment into the study, and it has genetic information. So one of the things that makes the UK Biobank such a powerful resource that the study that we're performing is that it has prospective data on all the participants. So the registry is linked up to the death registry, to cancer registry. We have information from hospital episode statistics of any interaction that the participants had with secondary care, and coming towards the end of 2018 should be linkage to primary care, and that will greatly expand the types of disorders that we can look at. And then some participants also participate in ongoing questionnaires through web interfaces. From our perspective of a genetic study, what makes the UK Biobank really special is that we have genome-wide genotyping on all participants. So this was carried out through a genome-wide chip that we use in many research studies. It had over 800,000 variants from across the genome. And so this captures both common variation we know is associated with disease status, and also some rare variants. Now this is genotype information, so it's sampling specific sites across the genome. But we do have, coming up shortly over the next few years, additional studies that look at sequencing, either exome sequencing to cover the protein coding regions of the genome, and then some participants will also get whole genome sequencing, so every single base pair of our 3 billion DNA base pairs. So the study that we carried out in UK Biobank was to predict morbidity and mortality using the baseline information and then using diagnosis of disease that we had from the registries and the linkage that I talked about on the previous slide. And so we're going to use the baseline information together with our genetic risk scores to see if we can effectively predict who's likely to get diagnosed with different disorders. So to give the actual details of the analysis that we performed in UK Biobank, we started with all individuals that we had genetic information and the right risk information for, and then we split those into two groups. Those that would be assigned to the standard risk category because they had no serious disorders at baseline, and then we pulled out those that would be in a non-standard risk group. So for the models, we used a Cox proportional hazard survival model. We took baseline information of standard factors that would be available for underwriting, such as family history, smoking status, and previous disorders. And then we added on the polygenic risk scores. So remember, this is a continuous variable that measures someone's liability to disease. And we looked at the predictive value of those polygenic risk scores. Our results for the genetic prediction of breast cancer, there's an incidence of just under 2%, so 2% of those women have gone on to develop breast cancer post-baseline. So in the table here, you can see in the left-hand column the percentiles of the polygenic risk score distribution. We're going to use as our baseline, as our reference group, those that lie between the 40 and 60 percentiles of this distribution. And the women that fall into the categories above the line are those that should be at reduced risk of breast cancer because they carry fewer genetic variants, and those that lie below the line are those that should be at increased risk because they carry more genetic variants. So the key question here is, does this polygenic risk score increase prediction of breast cancer? And does it increase it significantly of a magnitude that we would be interested in? Looking at the odds ratios here, you can see, as you expect, that the women above the line have a reduced risk of breast cancer, and that reduces to about a half-fold risk for the people in the top 10%. But importantly, for those below the line, their risk of breast cancer increases quite substantially. And you can see that the women in the top 10% of this distribution have a twofold increased risk of breast cancer. And that's pretty similar to what we might see for a positive family history. But recall that we have already removed family history by putting it into the predictive model. And then on the right-hand side, you can see the results for the standard group of women who had no morbidities, no major disorders at baseline, and their results are very similar here. Again, the polygenic risk score for breast cancer has about a twofold increased risk for women in the top percentiles of the distribution. For coronary artery disease, we performed exactly the same analysis. This time our baseline model included those risk factors that are relevant for coronary artery disease. So we had socioeconomic status, BMI, family history of relevant disorders, blood pressure and alcohol intake. And again, looking at the polygenic risk scores, you can see that they behave as we might expect, that those people in the categories above our reference guidelines have a reduced risk of coronary artery disease, and those below the line have an increased risk. The values here are slightly lower than we saw for breast cancer, so we only reach that twofold increased risk for the very top percentiles there. And then for those at standard risk, again, the results are similar, but they're slightly higher here. So the people in the top percentiles of risk have a higher risk of coronary artery disease than when we looked at the full cohort. And we think that that's probably because of our selection bias. So some people from this group who are already at high risk and are showing symptoms, high blood pressure, would not be included here, so they've been removed. So we've shown that looking both at breast cancer and coronary artery disease, that the polygenic risk scores, that our genetic predisposition to these disorders, does have a significant statistical contribution to risk prediction. And that's a contribution above and beyond what we would normally include as underwriting risk factors.
Thank you, Cathryn. That concludes the third of our webcasts. If you have any questions on this webcast or any other in the series, please email me at the email address shown on your screen now. We hope you will choose to view the other webcasts in the series, and they can be accessed at the website shown below. Thank you.